Good morning, student. Today we will learn about replication in DNA from the sixth chapter, that is molecular basis of inheritance. Okay, so in replication of the DNA, same DNA copies will be made. Okay, let's learn about central dogma of the life. That is conversion of DNA into DNA only. It is known as replication. DNA to RNA, that is known as transcription, and RNA to protein, that is known as translation. Today we will learn about replication of the DNA. So this is the DNA structure, as you all know about it. Whole DNA is like this. This is very super coiled structure, we can say. I am taking this much part. Okay, and in the replication you must know that whole DNA is replicated together in one process only. So the DNA will be divided into pieces. These pieces are known as replicone. So I am here showing you the replicone. Now, one enzyme, first process, that is an enzyme, that is known as helicase. What is the function of helicase over here? That it unwinds the DNA strand. So, unwinding of the DNA is done by helicase enzyme. Now, second, that is of SSB protein. Full form of SSB is single stranded binding protein. Over here, single stranded binding protein, the uncoiling of the DNA, maintaining of the uncoiled DNA is done by SSB protein. Over here, at the end of the uncoiled DNA, SSB proteins are situated. Third one, that is topoisomerase. Now, topoisomerase functions like it prevents the negative uncoiling over here. Okay. Prevent negative coiling at the end part. Now, let's start the process. If any unwound DNA will do replication, over here two partition will be made. One replication will start in this direction and second one will continue in this direction. So let's study the half of the part. This part where the replication start, it is known as replication fork. What it is known as? Replication fork. And this strand, it is known as origin of replication. Now you know that DNA is a double helix structure over here, 3 dash, 5 dash, and so replication always takes place in 5 dash to 3 dash direction. Remember that. Replication always takes place in 5 dash to 3 dash direction, but the main part of the DNA that is known as template where replication will take place. So that is known as template, and template direction should be 5 dash to 3 dash in opposite direction. Okay, so this is also a template. Both the strand will create a new DNA, and let's start the process. Over here, some primer. What is primer? Which help in the starting of the replication process. A small part of the nucleotide that is known as primer. So if the replication continues into 5 dash to 3 dash direction that you know, so the template should be what? 3 dash to 5 dash, then and then only replication will start at particular direction. So the primer will bind over here in this template strand, and in above template strand the primer will bind here. Why? Because here the replication will take place 5 dash to 3 dash in this direction, but over here replication will take place in 5 dash to 3 dash direction opposite to the that template. Okay, so primer will bind at 3 dash direction always, remember this thing. And over here a strand of the DNA synthesizing started, and over here this strand will synthesize a DNA strand. Now let's uncoil the other parts of the DNA. Over here primer is in this direction, and here primer is in this direction. So यहाँ पे इतनी स्ट्रैंड सिंथेसाइज हुए, यहाँ पे इतनी स्ट्रैंड सिंथेसाइज हुए, और ये यहाँ पे सिंथेसाइज होते जाएगा, बट over here we need to synthesize a new primer. So at the end what will happen, we will get over here the continuous strand. It is known as leading strand of the DNA replication, or it is known as leading strand. And over here small primer structure we are getting in between that, so it is known as lagging strand. Now this primer will be removed further, and on the DNA fragment we are getting small fragments of the DNA on the lagging strand and continuous DNA on the leading strand. Now by the help of DNA polymerase, which enzyme? DNA polymerase, these gaps are filling by the process of polymerization. And further the ligation of these fragments. These fragments first of all are known as Okazaki fragment. What it is known as? Okazaki fragments. And this will join by the enzyme ligase. Okay. So from first let's revise it again. DNA replication, that is DNA to DNA. Now the replication part, it is known as replicon. First enzyme, that is helicase, it will unwind the DNA. SSB protein, it will keep the unwinding of that particular DNA part. Topoisomerase, it will prevent the negative calling at the end of the part. And now where replication start, it is known as replication fork, and the DNA strand on which replication will start, it is known as origin of replication. Now on the main DNA, it is known as template strand, and always replication take place in the 5' to 3' direction over here. And we need a primer. What we need? A primer. It will bind at the 3' direction only, and on the leading strand, that is 3' to 5' direction strand, will synthesize 5' to 3' DNA, and on the lagging strand, the fragments we found, and those fragments are known as Okazaki fragments. Now those fragments fill the gap by with the help of DNA polymerase enzyme, and that process is known as polymerization. And the joining of the DNA fragments or Okazaki fragments has been done by ligase enzyme. Thank you.